Enzymes are awesome. Why? Because they can speed up reactions like hundreds of thousands of times, and they make things happen that are actually like specific and meaningful inside of a sea of all sorts of molecules just floating around. But how specifically do enzymes do this? They use a variety of different catalysis mechanisms, but these are kind of like conserved mechanisms that we see over and over and over and over again in different enzymes. And so if we could recognize those mechanisms, then we can kind of recognize the strategies enzymes take. And then it doesn't seem as overwhelming that we have all of these different enzymes and all these different reactions. Let's focus on those key things. Let's focus on those catalytic strategies as well as the main reaction types and how we can classify enzymes based on the reactions that they catalyze.
Today, we're going to continue on our discussion about enzymes and go in a little more depth into how enzymes exactly do what they do. We don't need to worry about the nitty-gritty details. I don't want you memorizing the specifics of each specific reaction, but instead we're going to focus on the kind of logic enzymes use. We'll see that there are a set handful of kind of strategies that all enzymes are going to employ, or that at least a lot of enzymes are going to employ, but they kind of mix and match different strategies. They can do this in order to catalyze a large variety of different reactions, while each enzyme itself is going to be highly specific. We'll talk about how we can kind of look at reactions, look at kind of like the before and after. And similar to how in those cartoons in the Sunday comics, you might have those pictures, it's like spot the difference. We'll practice spotting the difference between the substrate and the product and try to identify what sort of reaction has gone on and therefore what sort of enzyme might have been at play. We'll also practice at looking at reaction mechanisms and identifying the types of catalytic strategies that are being used. So let's start with just reviewing our enzymes and sort of those main catalytic strategies, but going a little more in depth this time. So remember that enzymes are going to lower the energy of the transition state. So their job is to lower the activation barrier. Remember, if you have your leprechaun and there's a pot of gold at the other side of the rainbow, but it has to get up the top of the rainbow in order to get to the pot of gold. Even if it would be much, much happier with that pot of gold, even if it's more thermodynamically favorable to be at that pot of gold, you have to get over the top of the rainbow. And if that rainbow is really, really tall, it's going to be hard to do that. So what an enzyme is going to do is it's going to lower the height of that rainbow. It's going to lower the energy of the activation barrier and allow the leprechaun to get to the other side. But when you're at the top of the rainbow, you can go either way. And so enzymes are going to catalyze the reaction in both directions. Remember that we can also kind of more formally talk about that transition state and the activation energy. We represent the transition state with like this double dagger symbol. And the energy taken to get to that transition state, the activation energy, we represent it with this delta G double dagger. So remember that the overall delta G is going to be the difference between the energy of your substrate and the energy of your products. But then what can vary, that's not going to change, but what can vary is the path in between them. And the height of the tallest peak, the highest transition state, that energy required to get to that delta G double dagger for that highest energy transition state, that's going to be our rate determining step. And so if an enzyme wants to speed up the rate at which an enzyme reaction happens, it's going to need to lower that activation barrier. And we saw how one of the key ways that enzymes could do this was by providing lots of favorable interactions to the transition state. So we wouldn't want it to bind too tightly to the substrate because then we're kind of digging ourselves a deeper hole that leprechaun has to go climb out of. But if we lower the transition state, if we provide these favorable binding interactions to the transition state, well, now we're able to lower the activation barrier, allow us to get to the other side more easily. So for example, if we had a nucleophilic addition and elimination to a carbonyl group, which we're going to see over and over and over again, what we would see is that basically we have a nucleophile. Remember, we practiced identifying our nucleophiles and electrophiles, and we said that often a nucleophile is going to be a lone pair and an oxygen or a nitrogen, and our electrophile is often going to be a carbonyl carbon. We draw our arrow from our nucleophile to the electrophile. So in this case, our nucleophile is going to be the oxygen on this water, and our electrophile is going to be this carbonyl carbon. When we have them attack, basically what's going to happen is ultimately we're going to form a tetrahedral intermediate. So an intermediate, we have actual bonds here, but in order to get that to that point, we first had to have a partial bond being formed and this partial bond being broken. So this is our transition states. This is what we put in brackets with the double dagger. This is what the enzyme is going to be the most stabilizing to and provide the most help for. Additionally, we would also have another transition state on this side of the intermediate as we go from having these bonds formed to then breaking off this other bond. In this example, they're only showing the transition state, this transition state, because this is going to be the slower one, the rate determining step. But you are also going to have a transition state on the other side, and so you're going to have two peaks on either side of that valley representing where the valley is your intermediate, and the peaks on either side are going to be your transition states. But in order to even get to this transition state, we have to make it more likely. And so we can do this in a couple of ways. And so we're going to look at how acid and base catalysis can either strengthen the nucleophile or strengthen the electrophile, and how other things like metals can help stabilize the charge on the transition state. And all these enzymes can kind of all provide interactions with this transition state that are going to help lower the energy required to actually get there. And then once it gets there, which way it goes is going to depend on the overall thermodynamic favorability of the reaction, which is something that that enzyme can't change. But by doing it in little steps where each step isn't quite as bad, it can do things like make it easier for the reaction to happen. And later in the course, we'll look at how by kind of controlling the concentrations of the products and the reactants, we can favor one direction or the other. So let's talk a little bit more about some of the main catalytic strategies that enzymes use. So the four main ones that we've mentioned, we're just going to review them briefly. We have proximity catalysis, also known as entropy reduction, acid-base catalysis, metal catalysis, and covalent catalysis. We'll review each of these and then look at some examples of how we can see them used and try to identify them in various reaction mechanisms. So pretty much all enzymes are going to use the strategy of entropy reduction or proximity catalysis. Basically, they're just going to help the molecules find one another and stay in the right orientation to react. So you imagine if you have these two molecules and you want them to join together. If they're all floating around, if one of them is in your bedroom and one of them is in Alaska, assuming you're not in the bedroom in Alaska, it's going to be really hard for those molecules to find one another. It's going to be hard for those things to find one another. One of the jobs of the enzyme is to actually kind of hold those things close together and hold them in the position so that they can interact. Remember how our free energy, we had that entropy component and we had that enthalpy component. And we said we wanted our entropy to be higher in our products and in our reactants. We want there to be more randomness, more disorder in order for something to be favorable. But if we're bringing two things together, well, we're kind of inherently having a loss of entropy for those two things in particular. But we can offset that loss of entropy by a couple of different things. We can make favorable reactions with that transition state, what we saw before with forming those interactions and lowering the transition state barrier. And we can also kick out water. By kicking out the water, we increase the entropy of the water, and therefore we're able to help compensate. But by kind of bringing the things together and holding them in the right orientation, we're kind of prepaying the cost of the loss with entropy with gains in this enthalpic component, with having those favorable interactions with the enzyme that we saw can actually, just like in one interaction with that transition state, can lead to a large decrease in our activation barrier. So we get a large amount of so-called binding energy. So pretty much all enzymes are going to use entropy catalysis, and a lot of them are going to use acid-base catalysis, but not all of them. So remember we had two types of acid-base catalysis, no pun intended. General acid-base catalysis is going to use acids and bases other than water, or at least they're going to use an acid or a base other than water to activate a water molecule. Whereas specific acid-base catalysis, these are going to use water ions as the acid or base, and they're going to rely only on the pH. So in the case of a specific acid or base catalysis, what we're saying is that it's only those hydroxyl ions and those hydronium ions, those protons in the solution that are just naturally in the solution because of the pH. Those are the ones that are going to be participating. Whereas with general acid base or catalysis, which is what we'll typically see with enzymes, we have another source. We have typically an amino acid residue in the active site that's actually going to be giving and taking protons. Sometimes we'll see that they'll give or take protons from water in that active site in order to activate the water, or sometimes they're going to actually interact directly with the substrate. But in both of those cases, you need more than just having the ions present in the pH. Due to the pH of the water, you need an external source that's coming from your enzyme itself or from a cofactor. So we looked at this example. We had water attacking a carbonyl carbon. Now, this would not be very fast because, well, water is not a very good nucleophile, and that carbonyl carbon isn't a strong enough electrophile unless we have a really good leaving group. And so we'll talk about how we can do things like add a phosphate in order to make a better leaving group. But for now, let's look from the perspective of our nucleophile and our electrophile. Remember, we need to, when we identify a nucleophile or an electrophile, we need to label it directly on the atom so that we know exactly what's participating, not just the whole molecule. So we could label this as our nucleophile and this is our electrophile. So as we said, this nucleophile isn't very strong and this electrophile isn't very strong. So how can we make them stronger? We can do this with acid and bases. In the case of a general base, here what we're going to do is we're going to take a proton in order to strengthen the nucleophile. So we're going to take a proton from that nucleophile. So we might take a proton from water, strengthen it. When you have a hydroxide ion, that's going to be a much better nucleophile than a water molecule, and therefore it's going to be better able to attack. When it attacks, basically what you're going to get is you would have this growing positive charge on this oxygen unless you stabilized it by kind of having this general base nearby that's abstracting the proton, preventing that positive charge from building up. Then you can have this reaction proceed more quickly than it would in the absence of that enzyme. What about a general acid? Well, we said that the general base could strengthen our nucleophile. The general acid can strengthen our electrophile. If we go and we protonate that the oxygen, what we're going to have is that we're going to make this a better electrophile. We're going to make it more vulnerable for attack from our nucleophile. Our nucleophile can then go and attack, and this is going to make for a better, a more favorable tetrahedral intermediate that can also then provide a better leaving group if we wanted to then kick this off and leave this group. When it comes to general acids and general bases, it can get confusing because I know we talked about like how the basic amino acids were our histidine and our lysine and our arginine, because in their neutral form, they acted as bases and took a proton, becoming positively charged. And we said that the acidic amino acids were going to be our aspartate and our glutamate because in their neutral form, they acted as acids, gave up a proton in order to become negatively charged. Well, here I'm going to tell you that those general acids, that in terms of general acids and general bases, those can both be general acids and general bases, all of those, as well as serine and tyrosine and cysteine. Basically anything that can act as like a nucleophile, anything that can give and take protons, all those things are going to be able to act as general acids and general bases. Yes, both a general acid and a general base. Because remember that enzymes, they're catalysts, they have to be able to do this over and over and over again. And so if they give a proton, in order to be able to give a proton again, they have to actually take that proton back. And so we're going to see that these amino acids are going to act as both acids and bases at different steps in a reaction. So your book has a lot of great information and examples. And I'll also post some links to some supplemental reading, such as this chapter on enzymes from LibreText. So LibreText is really, really great if you haven't discovered it already. It has a bunch of free texts. So we've kind of used some of them already. But you can also explore more of this library yourself. So here's one on enzymes, and it has a nice example of how an enzyme is helping to catalyze the methylation of DNA. So we'll look more later in the course about how DNA can be modified, such as with the addition of a methyl group, typically with an enzyme that's going to be using this cofactor called SAM. And so we'll get much more into cofactors and this sort of thing later. But for now, just see that we have this molecule called SAM, and it's got this methyl group, and it's going to be attached to the sulfur. And we want to transfer this methyl group from SAM onto the amine group of adenosine. In order to do this, we're going to kind of have to get this adenosine to attack this carbon and kick off this part. In order to do this, the enzyme is going to help out. So when you look at a diagram that's kind of showing how these enzymes help out, typically what you'll see is something like this. We're going to see those arrows that are going to represent the flow of electrons from our nucleophile to our electrophile. And then we're also going to see some of the amino acids in the enzyme, but not most of them. If we showed all of the amino acids, it would be like when we were looking in our 3D structures the other day, and it was kind of hard to make out what was what unless we use like SLAB or something in order to isolate specific regions at a time. So in the case of one of these reaction mechanisms, instead of showing all the different side chains, which will just clutter everything up, we only show the specific amino acids that are somehow involved, either in binding to the substrate of the transition state or to actually helping aid the catalysis, directly acting as catalytic residues. In this case, what we're looking at is it's showing us that we have an aspartate on this side, and on this side, we have a proline. So remember with proline, that's one of our weirdos. It's got this side chain that kind of loops back on itself. And so what we're looking at here is going to be the carbonyl oxygen on the actual backbone of the proline. Whereas here, we're looking at the aspartate side chain. What we can see is that carbonyl oxygen, remember that usually that's going to be like in the secondary structure, that would be forming a hydrogen bond to one of the amino groups on another chain. And remember also we talked about how proline, it doesn't have that acceptor hydrogen on its nitrogen because it's kind of taken up with the side chain. But that carbonyl oxygen can still contribute to hydrogen bonding. And in this case, it's going to be hydrogen bonding to this amine group on our DNA. Why is this important? Well, this is important because that amine group is going to be having to do the attacking. And in order to make it a stronger nucleophile, we're actually going to make it negatively charged. We're going to make it negatively charged by stealing a proton from it. So this aspartate can steal a proton from this amine group, make it negatively charged and more nucleophilic. But in order to kind of like make it negatively charged, well, that's not going to be very favorable. That's going to have a high activation barrier. But by forming a hydrogen bond on this side, well, now you're able to lower the energy of that awkward transition state of that transition. And therefore, you're going to make it easier for this to happen. So by forming this hydrogen bond, you're helping stabilize this. When you're stabilizing this, you're making it easier for this to attack. And so this is showing us how we can have these interactions, both these sort of like direct catalytic reactions where we're acting as a general base, but we can also have more of these just like stabilizing interactions from additional amino acids, both from their side chains as well as from the backbone. So all of these can contribute to ways in which the enzyme is able to help make this reaction happen. In these cases, we're just kind of giving and taking protons. And remember, we give and take a proton, we're not actually making a new like covalent bond to anything other than a proton. We're not linking two molecules together. We're just adding or removing a proton. But sometimes we do see that enzymes will actually form covalent bonds, will actually link directly to the substrate, attach themselves to the substrate as an intermediate. And we call that covalent catalysis. We'll look at some examples of this later, such as a serine protease, in which our enzyme is actually forming a covalent intermediate with the substrate itself. And finally, we have metal catalysis. Metals are really awesome. They can do things like stabilize negative charge in that transition state, helping lower that activation barrier. We'll see that they can give and take electrons to help with redox catalysis. So we'll see this a lot when we go into metabolism next semester. And we'll also see how they can kind of like form coordinate bonds. So basically, they form these dative coordinate bonds where a pair of electrons, such as on a nitrogen or on an oxygen, similarly to how we saw in our case of hemoglobin, they were basically forming coordinate bonds with the metal. And the metal has this big electron cloud. It has this diffuse positive charge. It's able to form these coordinate bonds with a number of different atoms at once. And this is going to allow that metal to serve as a central coordinating hub. And it's also really great if you have a reaction that's going to use like ATP as a cofactor. ATP has that negative charge. The positive charge of your metal cation is going to help stabilize that and help make this reaction more favorable, and even do things like make the phosphorus in a phosphate group even more electrophilic by pulling the negative charge even further away. And so we'll look at how we can find examples of each of these different things at play. And often we're going to find multiple of these strategies at play within a single enzyme. So let's look at some examples of how enzymes mix and match these strategies in order to catalyze reactions. Here's one example, phosphorylation. So remember our good old phosphorylation? What is it that adds a phosphate group? Right, our kinases. Our kinases are going to be the thing that's going to add a phosphate group. And they're typically going to add it to a serine, a threonine, or a tyrosine on the hydroxyl group. The phosphate group, or more technically the phosphoryl group, so remember that the oxygen is the one that comes from the protein, and then this phosphoryl group, these phosphorus and these three oxygens are going to be what's being transferred. And they're going to be transferred from ATP. This is going to leave us with ADP and a phosphorylated protein, or some kinases are going to phosphorylate small molecules. And we'll see this a lot when it comes to metabolic pathways, that phosphorylating those small molecules is a great way to do things like make good leaving groups. But for now, we're just going to look at this example where we're phosphorylating a protein. In this case, it's the protein itself that's actually going to have to act as a nucleophile and attack the ATP. The job of the enzyme is to make it easier for that protein, for that hydroxyl group on that serine or on that threonine or on that tyrosine to actually go and attack the ATP. What we're going to see is that in the active site of that enzyme, we're going to have a hydroxylate. We're going to have this negatively charged oxygen that's going to be able to act as a general base, abstract a proton from our serine or threonine or tyrosine, from our protein substrate, in order to make that a stronger nucleophile. And now the stronger nucleophile can go and attack that electrophilic phosphorus in our ATP. But in order to do this, well, we need a couple things. We need that ATP to be there, we need it to be in the right position, and we need that phosphorus to be particularly attacky. And so what we're going to do is we're going to use magnesium in order to help with this. Magnesium, remember, it's going to have this positive 2 charge. It's going to be able to bind to those ATPs and stabilize it. It's going to stabilize the transition state when we have this attack forming. It's going to stabilize the negative charges. It's going to pull electrons away from those phosphorus. So remember that phosphorus is going to be electrophilic to begin with because the oxygens are pulling electrons away. And now we have the magnesium pulling electrons away from the oxygen, which were already pulling electrons away from the phosphorus. So now this phosphorus is going to be especially electrophilic and vulnerable for attack. We've used this general base to make a stronger nucleophile that then is going to be able to attack. When we have this attack happen, now what we're going to get is we're going to get the ADP pushed off. This is an SN2-like reaction, so it's going to happen in this concerted motion. We have the oxygen attack the phosphorus, and then we get the ADP kicked off. Before this ADP leaves, it can pick up a proton from that hydroxyl group that was serving as our general base in the beginning. So now remember it has this proton, so it can't act as a general base again until it gives it back up, until it acts as a general acid. So it can donate that proton back to the ADP, make the ADP more stable as a leaving group by stabilizing the negative charge, and it can also then regenerate itself to do this reaction again. Have the ability to act as a general base again. Speaking of that, what catalytic strategies are being used? So we just gave away the one that it's starting by acting as a general base. And then at the end, we saw it acted as a general acid. But what was it doing? What else do we see go on in this reaction? Right, we saw metal catalysis. We saw that magnesium sort of serving as that hub and stabilizing the negative charges, helping coordinate things. And we also saw proximity catalysis. We saw that the enzyme was helping hold things in the right orientation to react. So remember proximity catalysis, that's also known as entropy reduction. And we're basically going to see that with pretty much all the enzymes. One of the things we didn't see though, one of the things that's less common is covalent catalysis, where we're actually forming a covalent intermediate in which the enzyme is attached to the substrate, or at least part of the substrate. And so we'll look at some examples of this later, but that was not at play here. We saw that enzymes can kind of mix and match strategies in order to catalyze reactions, and enzymes are typically going to specialize in catalyzing one reaction one way. But if we compare enzymes, well, we can see that different enzymes can catalyze different reactions, and they might do so in similar ways. Enzyme 1 might catalyze a different reaction than enzyme 2, but they might use similar mechanisms. On the other side, you could have enzyme 1 and enzyme 2 basically be catalyzing the same overall reaction, but doing so in different ways. So we're going to see a lot of versatility in enzymes.
Okay, so let's look at a couple of examples involving proteases or peptidases. So let's look in a little more detail about our serine proteases. Basically, it has this cool like catalytic triad. So triad, there's 3 catalytic residues. We've got a serine, a histidine, and an aspartate. And what's going to happen is we're going to get this oxygen to act as a nucleophile and attack the carbonyl carbon. But the serine, remember, isn't going to be a very good nucleophile as is. Because remember that the serine is not normally going to be deprotonated because it's got this high pKa of about 13. So we need to actually lower that pKa. So that would be the pKa in isolation, but in the context of the active site, we can actually lower this. And so how do we lower the pKa? Well, we lower the pKa by helping stabilize the negative charge, helping pull away the proton. These things would lower our pKa. And so we're going to find that we're going to have serine get its proton stolen by histidine, and histidine is going to help make more basic, more wanting to take that proton by an aspartate being nearby. So what we have is basically you have the pocket on the enzyme in the active site where there's like a substrate binding pot. And this is binding there is going to get that trio into position to get to work. So you wouldn't want things always to be activated, always on all the time. Instead, you only want to activate it when you have a substrate bind. So when the right substrate binds, it's going to fit nicely in the pocket of that enzyme. And the enzyme, remember, what's it going to stabilize? It wants to stabilize the transition state. And so basically, you're going to have like that induced fit. The induced fit is going to help make it so that that trio is right in position to get to work. So the first things first, we got to pull off that proton from serine. And so how are we going to do this? We're going to do this using a histidine. But histidine, well, that's not a very good base, remember? And so how are we going to get histidine to be a good enough base to pull off a proton from something that's not a very good acid? This is where the aspartate comes in, because remember, aspartic acid is a great acid. It really wants to be in this conjugate base form. It really wants to be in this negative form. In this form, it can hydrogen bond with this, with one of the amines in the histidine. By forming this hydrogen bond, what's going to happen is that when the histidine gets protonated, it's better able to stabilize that charge. So it has this kind of like negative charge that can help make it stabilize this protonated form. And so the histidine is then going to be able to take that proton. Once you've taken the proton, well, now you've got a strong negative. You've got a strong nucleophile. That serine really doesn't want to be negative, so it's going to attack that carbonyl carbon. Now remember that the enzyme is helping everything be held in the right orientation to do this. It'll attack the carbonyl carbon. We get our tetrahedral intermediates. When we attack a carbonyl carbon, we get a tetrahedral intermediate. It is a real intermediate. It's not one of those transition states, but we did have a transition state in order to get here. So we had to form a bond to the carbon, and then we had to break this bond, this double bond, so to get to this tetrahedral intermediate. So in between those, we would have had a transition state where we would have those partial bond formations. But
now what we have is basically we have this tetrahedral intermediate. And it's attached to our substrate in this case is covalently attached to the enzyme because the serine is coming from the enzyme. This is our substrate, but we haven't broken our substrate yet. We've only broken one of those double bonds. And so now we need to break this off. And we want to make sure that we break it off the right way too. The enzyme is going to help in a couple different ways. Basically, there's this part on the enzyme, this called this like oxyanion hole. So oxyanion, because it has this negatively charged oxygen. And then there's going to be the amide nitrogens in the backbone of the peptide in this pocket are going to help stabilize this intermediate and kind of make sure that it's going to break the right way. So this intermediate is going to break down. What's going to happen is that the nitrogen in what was the peptide bond is going to steal a proton from that histidine. And so this is going to be stealing this proton. This is going to break the first part off, but the N-terminal part is still stuck on the other part. So you still have part that's stuck to the enzyme, and then you have part that has been broken off. So we need to, in order to use this enzyme again, we need to regenerate it. We need to basically kick off this other part so that we can get this, to do things over again. What are we going to call this? Right, this is going to be one of those general acid-base reactions. And in this case, we're going to be having a general base because this nitrogen is going to be stealing a proton, making this hydroxyl ion that can then go and attack the carbonyl carbon. You get the whole situation happening again, but this time, because you're doing it from water and not doing it from the amino acid, you're going to be breaking things off. And so you're releasing the rest of your product. And you can see that we have kind of like two transition states that are pretty similar because we have to do that like reaction state twice. So what types of catalysis was that an example of? Right. So that was an example of acid-base catalysis. That was an example of proximity catalysis or entropy reduction. Not a metal catalysis because we didn't have any metals involved, but it was a covalent catalysis because we were forming that covalent intermediate. But that's not always going to be the case. So with some proteases, they're actually going to not form a covalent intermediate. An example of this is going to be our aspartic proteases. In aspartic proteases, here, they're just going to make water do all the work from the beginning. In the active site of an aspartic protease, instead of having our catalytic trio, we have two aspartate residues. So remember our aspartate, it's got that really low pKa normally, and it's going to want to be deprotonated. So it's already in that conjugate base form that can ready to act as a general base to steal a proton from water, make that water a stronger nucleophile, and go attack. But because it's going to be, aspartate is going to be a stronger base, it's not going to be very eager to steal that proton. However, by having this other aspartate nearby, well, now this is actually going to raise the pKa of the first aspartate, make it so that the first aspartate is more willing to take a proton. Having these two negative charges next to one another with negative charges repelling, it's going to seem more attractive to this aspartate in order to actually go and take a proton, to get some positive charge, to balance things out and help it not be as repelled. So this aspartate can actually have a higher pKa where it'll take that proton and now it'll go and attack. Once it attacks, basically the water is doing the attacking, so you're not forming a covalent intermediate, but you are going to still be having this water go and break the bond, and ultimately you get your peptide bond cleaved. So remember that basically what we can see is that we have the pKa can be dramatically altered in the active site of an enzyme. And so that's why I was using that caution when we were looking at those pKa's in those tables and saying, it's going to be different in an actual protein, and especially in a specific site. Those pKa's might be very different than they would be in the natural environment if they were just hanging out in water as free-floating amino acids. So we see that by kind of manipulating what's around it, we can manipulate the pKa of different amino acid residues, make them more or less eager to act as an acid or act as a base, act as a nucleophile, whatever the case. So that would be an example of an aspartic protease. And in this case, we could also do like draw out a reaction diagram, and we have our transition states and this sort of thing. Remember that the intermediates are going to be our valleys, and then our transition states are going to be the peaks. And you can see that you can kind of quickly get into these more complicated transition state diagrams, but the one that has the highest free energy change is going to be the one that's going to be rate determining. So we can see that the first attack of the water is going to be the rate determining step, but the aspartates are going to make this so that this is actually possible. Here's a real life example where we can actually see this in play. So this is showing you the active site of pepsin, which is one of the aspartic proteases. We can see the aspartates in the active site that are going to help make this reaction happen. And here's the active site of an HIV protease. So you can see here, you kind of have these two different subunits, and each of them is contributing an aspartate that together make up this active site that's able to carry out this proteolysis. And so HIV protease is an example of an aspartic protease. So what type of catalysis did we just see? Yeah, we saw acid-base catalysis, we saw proximity catalysis or entropy reduction. No renal catalysis, and this time we didn't even have that covalent catalysis. So we saw that different enzymes could catalyze the same overall reaction in different ways. We could cut a peptide either with a serine protease, in which case we were using that covalent catalysis strategy. Similarly, we could use a cysteine protease, which basically does the same thing except with a cysteine instead of a serine. Or we could use an aspartic protease, which is actually going to use these aspartates to make the water react. And there are even additional other types of proteases we could have, where we could have metalloproteases that are going to use metal, glutamic proteases, which are similar to aspartic proteases, but they use glutamate instead of aspartate. All of these are just different ways in which we can cut an enzyme, or we can cut a peptide using different enzymes. We can also have enzymes within the same specific types. So we can have serine proteases, and for example, that are going to catalyze different reactions. And when we talk about different reactions, all of them are overall proteolysis reactions, but they have different substrate specificities. So remember that when we're cutting off our tag, our GST from our protein, we're using the site-specific protease. We don't want a protease that's going to be super generic and just cut up our protein that we just work so hard to purify. Instead, we just wanted to cut specifically where we have that place, that site in between our tag and our protein, so we can cut our protein off from the tag, but we don't want any cutting anywhere else. So we need a protease that's going to recognize a specific site. And we'll find that some proteases have really high specificity, like those ones that we use to cut off our tag, like that HRV3C or that precision protease. Remember, that's the same thing, it's just a brand name. Or we can have proteases that are going to be kind of like more generic. And those generic ones are going to be good for things like digestion, where you want to kind of like break down everything. So we have enzymes like trypsin and chymotrypsin and these sorts of things that are going to help be more kind of generic, but even these are going to have some specificity. Serine proteases get their specificity thanks to this thing called the specificity pocket that we had talked about, or the S1 pocket, that's kind of going to help orient things in place. So if we look at 3 examples of serine proteases, trypsin, chymotrypsin, and elastase. Trypsin, it cuts next to basic residues. So it cuts next to long basic residues, so lysine or arginine. Chymotrypsin, it cuts next to these bigger hydrophobic residues. And elastase, it cuts next to these small residues. If we go and look at the specificity pocket, which remember is where the substrate needs to bind in order to kind of get that catalytic trio into position to react, here we can see that these specificity pockets are actually going to be shaped to kind of recognize the things that we're going to cut next to. So in the case of trypsin, you can see this little kind of like red dot right here. This is going to be representing this kind of like acidic pocket. That is where the trypsin specificity pocket is. That's going to nicely complement this lysine or this arginine. So you have this kind of long, narrow pocket with a negative charge at the bottom that's going to be perfect for holding on to one of those lysines or arginines. In the case of a chymotrypsin, here we want something that's going to be neutral, but we also need it to be big. So here with chymotrypsin, you basically see this big broad channel that's going to be hydrophobic. And in the case of elastase, well, here we have this little channel and it's got these valines sticking out of it so that it can really only accept these really little things like a serine, an alanine, or a glycine. And in this way, as well as with other reactions around here, we're able to kind of have specificity for a various substrate and not be just cutting anything. Although there's a whole variety of different specific mechanisms, we can kind of break things up into a number of classic reaction types. And these are roughly classified by their enzyme commission numbers or their EC numbers. So remember when we were playing around in Expazy and it was that exercise where we had to go and we had to look up what is this EC number for this enzyme and what do those numbers represent? Remember, each of those numbers was telling us something about the enzyme. And the first number in that EC number is going to be the main class of those enzymes. And we have these 7 main classes, but for the purposes of this class and things, instead of actually focusing on the exact number of these different classes, which by the way, I learned a great acronym, mnemonic, to have lived in London, which I'll tell you in a second. But basically, these are the basic types of reactions that we're going to see. We're going to see ligation carried out by ligases, we're joining things together. Hydrolysis, this is going to be carried out by hydrolases, we're breaking things with water. We have group transfer, which are going to be carried out by transferases. We're transferring groups, as it sounds like. Isomerization, so isomerases are kind of going to swap things around. Redox reaction are going to be carried out by our oxidoreductases. And elimination and our bond breaking in other ways are going to be carried out by our lyases. So now let's look at a little more explicitly at these. So as I said, O2 have lived in London. And then they had this this last class added these translocases. This is actually this really new class. So there had been 6 classes traditionally, and then they're like, hey, there's actually a 7th class. And so now there's translocases, which are enzymes that move things like ions, often across a membrane. And we're not going to worry too much about the translocases, and you still won't see them referenced in many sources. But we will see these other types, and the reason why it can be helpful to know the numbers. And so I don't really care too much that you know, oh yeah, 4 is a lyase, except that when you go and you see an enzyme commission number, if you see a 4 and then you're like, oh, to have lived, and you're like, oh, lyase, then you can kind of tell what's going on. But that being said, what I care most about is that you're able to go and recognize what's actually happening in these reactions and why we classify something as a lyase or why we classify something as a hydrolase or a transferase. So let's go one through one in these different types of enzymes. In the case of an oxidoreductase, this is going to catalyze an oxidation or a reduction reaction. So remember our oil rig, oxidation is loss of electrons or electron density, and reduction is gain of electrons or electron density. Thankfully, with enzymes, the names often give away what those type of enzymes they are. And so we'll typically see words like oxidases, dehydrogenases, oxygenases, or peroxidases. You don't need to worry about all these details about how we kind of like differentiate between an oxidase and a dehydrogenase or anything like that at this point. This is just to point out some of the common words that you'll see, some of the names that you'll see associated with the oxidoreductases. For now, what I care about is that you're able to recognize when oxidation and reduction are occurring. Next, we have our transferases, and what they're going to do is they're going to transfer functional groups. So one of the most common things we're going to see is our phosphate or our phosphoryl group. We'll also see things like a methyl group. We'll see things like amino groups. And so all of these are going to be examples of a transferase, where we're transferring a group from one thing to another. So our good old kinase, our favorite enzyme in the world, that's going to be an example of a transferase. We have a group transfer reaction in which we're transferring that phosphoryl group. Next, we have a hydrolase. A hydrolase is going to break bonds using water. Break bonds using water. So it's going to catalyze a hydrolysis reaction. We have some common examples of this are going to be our phosphatases, which are going to kind of undo the work of a kinase. We have phosphodiesterase, so this would be things like our endonucleases, our restriction enzymes. So an example would be our restriction enzymes. And we have our proteases that are going to cleave proteins. So remember in our lab how we cleaved our tag off of our protein? And what was the protease that we used to do that? Right, we use that precision protease, that HRB3C. So the protease is an example of a hydrolase. Okay, we have lyases. Basically, a lyase is going to make or break bonds without using water or without using redox. And so these are going to be a little harder to identify, but often what they're going to be doing is they're going to involve the formation and or the breaking of double bonds. And so we're going to see things like elimination reactions. So we'll point out some more examples of these, but know that this is kind of like one of the trickier things to actually recognize. An isomerase, this is one of the easiest ones to recognize. Here, we basically just have to see where things are being swapped around in a molecule. When we talk about swapping it around, we're not changing the actual atomic composition. We're not changing the number of each atom, but we're just changing their arrangement. This could be just their arrangement in like 3D space, in which case we could have like stereoisomers that are kind of like our left and right hand. And that would be an example of a racemase that's actually doing that sort of conversion. One of the examples we've seen is going to be with the proline. Remember how proline could be that cis or trans conformation in its backbone? Well, a cis proline enzyme can actually go and convert between those two. Remember that when you have one of those configurational changes and not just a conformational change, you actually need help to make and break bonds in order to do that. And so the enzymes can help with this. A mutase is actually going to shift groups around within a molecule and change what links where. And so we'll see some examples of both of these things at play. A ligase, here they're going to join two molecules together, typically with the use of ATP. Join two molecules together. Typically with the use of ATP. You might also see other NTPs, so you might see like GTP or something like this, and this would be an example of a ligase. So the ATP is often used to kind of like pay the cost of putting them together by making like pre-activating things as making better leaving groups. But the ATP itself is not really being joined onto the molecule like we saw. It's not being the ATP that's being transferred in this step. It's actually going to be the molecules being joined together, but using the ATP as energy. And so we'll see some examples of this. And then finally, we have those translocases, which we're not really going to worry about too much in terms of their classifications. But let's look at how we can go and we can actually identify what type of reaction was going on. First of all, let's think back to our kinase example that we were looking at. We said that that was an example of our acid-base catalysis, our metal catalysis, and our proximity catalysis. But what type of reaction was it overall? Right, that was going to be a group transfer reaction. We were doing a group transfer, transferring that phosphoryl group from the ATP onto our protein. Therefore, the enzyme, the kinase, we can classify it as a transferase. So now that we have a better idea about the basic catalytic strategies, so we've got our acid-base catalysis, we've got our entropy reduction or our proximity catalysis, which pretty much all enzymes use in order to kind of form bonds with the substrate, or more specifically with that transition state, that are going to lower that activation barrier, bring things in an orientation and place in order to react, kind of offset the cost of the loss of entropy it takes to bring those things together. We also had, with some enzymes, we had our metal catalysis, where we had a metal ion that was actually helping like stabilize negative charges and kind of serving as that central coordinating hub, maybe making things more electrophilic. And then we had our covalent catalysis, where we saw that the enzymes could actually make a covalent intermediate linked onto the substrate or part of the substrate. We saw that enzymes could basically mix and match these strategies, but did so in order to carry out a sort of distinct set of different types of reactions. And we looked at how we can kind of classify enzymes based on their reaction type. In class, we're going to get a little more practice, look a little more in detail about how general acids and general bases can speed up reactions, identify different types of reactions, and really kind of try to interpret the logic of enzymatic reactions. In the coming weeks, we'll go into some more specific examples and see how enzymes are able to be so super duper awesome.